For our project, we decided to make a video focused on the structure, functions, and history of DNA. We'll start from when the idea of DNA first came to light. On February 28, 1953, James Watson and Francis Crick walked into a bar and boasted to the world that they had found the secret of life. They are, of course, speaking of the discovery of, you guessed it, Deoxyribonucleic Acid, DNA. Specifically, the two were talking about their discovery of the double helix structure. Quickly leading to the topic of the structure of DNA. DNA makes up approximately all living organisms on Earth. The structure of DNA is a fascinating one. It is composed of two strands spiraling symmetrically parallel to each other. The double strand consists of the sugar-phosphate backbone. The two are held together by nitrogenous base pairs which are linked through hydrogen bonds. The two strands have the potential of splitting, and when they do so, each acts as a template for another complementary strand to be made, thus creating two identical copies of the original double helix of DNA. Imagine two puzzle pieces that are supposed to go together, but the pieces are complementary to each other and each is specifically made to fit the other piece. But now, once you take these two pieces apart, each is made another complementary piece that fits it perfectly. This is the process of how DNA replication works every time an organism reproduces. Moving on. The genetic code is made up of a series of only 4 different bases. Each different type of base is characterized by one of 4 letters, A, T, G, and C. These stand for adenine, thymine, guanine, and cytosine. Each can only be paired with one other lettered base. Thymine and adenine can only bind together, and as one would expect, cytosine and guanine can only bind together. These matched bases dictate the complementary strands being produced. When replicating, the nitrogenous base pairs are bonded together with almost no error. In terms of the length of human DNA, the succession of bases can seem to go on infinitely. To be exact, there are approximately 3 billion base pairs in the human genome. That's a lot of letters. Even though the rate of error is almost infinitesimally small, with a genome almost infinitely large, these errors can add up. These mistakes are called point mutations, and when these types of mutations become large enough in number, they can be responsible for certain kinds of diseases. Certain scary diseases. These types of mutations can also occur through generations. Cell division is necessary in both forming a female embryo and forming sperm, and because the longer the life of the male, the more rounds of sperm production, which causes more mutation. So that's fun. Also, even though there are more mutations for an offspring with older parents, there are still hundreds of new mutations in the offspring with younger ones, but they're mostly the not-so-harmful mutations. So, it's also important to note that our DNA is largely responsible for the uniqueness of every human being to one another. Each person has a specific and unique genome in which their genetic code makes them who they are physically. On average, humans differ by 1 letter every 1,000 letters. These differences in the genome are considered SNPs, single nucleotide polymorphisms. Fitting name, right? This point always brings me back to the obvious example of the snowflake, a beautifully designed piece of art that nature has produced with no exact copies in the world. The human genome is beautiful, isn't it? Though we are a unique species, there is some relation to all life on Earth for humans. That's right, any animal you can think of. And the measure of the relation between us and other organisms can be found through the comparison of DNA sequences. The more similar the genome is in terms of letters, letter sequences, etc., the more closely related we are to that organism. So to say we can see where we stand in terms of common ancestry to others. Not to say we're, quote, animals by any means, sort of. Anyways, that's not to say we can compare our relation directly based on the similarity of an organism's genome. For instance, did you know that we share 50% of our genome sequence with bananas? But that doesn't exactly mean we're half banana. Just food for thought. Now let's go back to the two that started all this. When Watson and Crick's structure was first brought to light, there was a lot of question about its accuracy. Crick attempted to figure out the reading frame questions with the base pairs. After much thought, he found a way that seemed right, but wasn't. Gradually, as research techniques grew more and more complex, the true codon reading frame was finally discovered. The new system was not as mathematically satisfying as the one drafted by Crick, though. But the new method found that some amino acids are coded for 6 codons, while other amino acids are only coded for 1 or 2 codons. Each codon was found to have a use. For example, there are 3 codons that code for the stop of the synthesis process. Unlike a lot of things in science, this new method had no order, no beauty. There did not seem to be any reason as to why this method works the way it does. No real strong chemical or physical affiliations between amino acids and certain codons. But the new method was such a specific and delicate balance that changing one base pair would be just like changing a letter of the entire alphabet. There would be no consistency and the whole thing would fall apart. The fact that this mechanism is seemingly 1 in a million further supported the idea that life was created at only one instance in the timeline of the Earth, almost like an accident. But if life on Earth was created as an accident, then why does all of life have the same mechanism for going from DNA to proteins? Crick proposed that life came to Earth as an infection. He said the bacteria was seeded into the Earth by some intelligent extraterrestrial beings. This didn't really catch on though. But the thing is that the mechanism did not have to arise because it outcompeted the other mechanisms. The fact that there is only one code used for all of life could have come from the freak accident of an asteroid strike. All the other mechanisms died out from the strike, but the mechanism used in life today was lucky enough to survive. Since the 1980s, we found that the code is neither frozen or accidental. If DNA arose as an accident in an ancient mix of materials, the DNA would have come from the chemical and physical processes going on in that mixture. This did not become the new predominant theory of the time, but there was enough logic and truth to it that scientists were interested to see whether it turned out to be confounding or not. The patterns that have been noted so far are patterns in the position within a codon and the base pair used at that position. For the first position in a codon, there is a pattern between the base pair used and the precursor used to make the amino acids. For example, all amino acids formed by pyruvate are coded by codons with the first letter T. The second letter of the codon is based on whether or not the amino acid is soluble in water or not. It is based on a spectrum. Some amino acids are more hydrophobic than others, while other amino acids are more hydrophilic than others. There are 6 amino acids considered to be the most hydrophobic. 5/6 of the most hydrophobic amino acids have T as the middle base, but all the most hydrophilic amino acids have the second letter as A, G, and C, and were not seen to have any major effect in the second position of the codon. The pattern with the third letter is not as deterministic. There are 8 amino acids that are to be considered fourfold degenerate, meaning that any of the 4 base pairs can be in the third position and the same amino acid will be formed. For example, GGG can be switched to GGA or GGC or GGT and still code for glycine. This makes sense because the first 2 spots on the codon are deterministic, meaning they are strict in what letter needs to be there. The third position, however, is not as strict and has more room for play. Now, let's figure out where RNA comes from. It wasn't a main concern of early research as people were more interested on the function of RNA, but they came up with many different hypotheses and debated with one another, like kids trying to prove who's right and wrong. Maybe it came from cyanide from outer space. Maybe it was made by lightning striking methane and ammonia. Maybe it was formed from a volcano. Who knows? Well, apparently when there's large numbers of RNA nucleotides, they condense spontaneously into long chains. But if there's not enough of them, RNA breaks down back into its constituent nucleotides. The problem is that every time an RNA replicates itself, it uses up nucleotides, which lowers their concentration. So where do you find a constant supply of RNA? Vents. They grow in vents. That nucleotides accumulate to extreme levels in vents due to the presence of strong thermal gradients. Thermal gradients produce 2 types of currents, convective currents and thermal diffusion, that circulate through these pores. These 2 thermal currents gradually silt up the lower pores with many small molecules including nucleotides. The concentration of nucleotide could reach up to 1,000,000 times the starting level. That's not all. Longer RNA and DNA molecules could accumulate to even higher levels compared to single nucleotides as their bigger size allowed them to silt up the pores more easily, so the more the merrier. DNA molecules of 100 bases are predicted to accumulate up to 1,000,000 billion times the starting concentration. Lesson learnt, having more friends is always better. And with such high concentration, all the binding of RNA molecules and stuff could fall into place. So firstly, thermal gradients should concentrate single nucleotides to extreme levels in vents, promoting the formation of RNA. Then these same gradients should concentrate RNA, fostering physical interactions between molecules. And finally, the oscillating temperature should promote RNA replication. There's no better way for this to work. So how do RNA go from replicating to coding for proteins? Well, it's the vents again. If you put RNA in a test tube with raw materials and energy like ATP, it will replicate, and not just replicate, but it will evolve. Okay, so RNA will get faster and faster at replicating until it reaches a monstrous level of efficiency known as Spiegelman's monster. But this monster doesn't get more complex as it ends up as a stretch of 50 letters, which is the binding sequence for the replicase enzyme, and without it, the strand couldn't replicate at all. The noble RNA chooses to sacrifice its own replication code for protein so that it could be part of the high-level selection, part of the larger entity. Now let's shift gears for our last topic. So you may wonder, how hard is it to invent DNA? Not that hard, probably. It would have formed as spontaneously in the vents as RNA, just to make sure the coded message retained and make an exact copy of the sequence of letters in RNA in the form of DNA. To convert RNA to DNA, you only need reverse transcriptase, which is mainly associated with retroviruses. Don't ever underestimate the power of a little tiny retrovirus. And here we're only beginning to scratch the surface of the makeup of a logical bedtime story for everyone, even if it doesn't really seem like a bedtime story. And if it doesn't sound convincing enough, you're welcome to take on the mission to continue uncovering the secrets of life.